Hi, yeah, my name is Etienne Exantus. I'm a rising senior at Indica High School, and we're researching receptor pathways and their role in graft compatibility at the Margaret Frank Labs here in Cornell. So, plant grafting is the process of reconnecting the vasculature of two different plants, usually done between plants of the same species, but can be done between plants of different species. We are looking into tomato pepper grafts, and our tomato pepper grafts were very incompatible, very unhappy. They all died. But one of the things we saw at their graft junction is that there was a lot of cell death, and that cell death looked a lot like programmed cell death, which is basically a plant's immune response. And so it's caused by this thing, which is a signaling cascade, and it has plants have receptors on their cell membrane, which pick up on chemicals usually produced by pathogens, and can trigger this hypersensitive response and cell death at the graft junction. Well, usually not graft junction, but usually site of infection. Um, but a previous member of the lab, Hannah Ray Thomas, gave us this data showing us that incompatible tomato pepper grafts have a huge overlap in upregulated genes, as in infected plants. And so that led us to our hypothesis that the tomato graft incompatibility that we're seeing is caused by hypersensitive response, which would cause the programmed cell death at the graft junction. So we decided to approach the question of what exactly is causing this hypersensitive response from two different places, from the top of our signaling cascade and the bottom. From the top, it would be the ligands, which would trigger this signaling cascade. But because there's so many receptors on the cell membrane, which all connect to the same signaling cascade via the immune hub, you have to really hit the nail on the head and find that one ligand responsible. So we received a list of possible ligand candidates that could be responsible for this reaction, and we decided to test it with a normally compatible graft to see if that new compatible graft would then become incompatible. However, the medias that we used to suspend the ligands in the middle of the graft were not very successful, and our lanolin and Vaseline that we used kind of destroyed our graft junction. So we're gonna have to explore using other media in the future. So we switched up gears and we looked at the bottom of our signaling cascade, which is the transcription factor, which goes along the plant genome and upregulates the gene responsible for programmed cell death and hypersensitive response. And so there's a family of transcription factor called ERF, and there's one specifically called PTI5, which is highly researched and proven to be related to hypersensitive response in all kinds of plants. And so we took a knockout mutant of PTI5. So hopefully the PTI5 at the bottom of the signaling cascade is gone and would shut off that signaling cascade, meaning that the plant's response would be turned off. So we took a PTI5 strain of tomato and grafted it with PTOR, which is our wild type, and pepper, hoping that we would have compatible grafts due to that. But unfortunately, we had really low survivorship in our grafts. A lot of them got infected with the fungi, and so we didn't get the results that we wanted. In the surviving grafts that we did have, it looked like there was very little to no interaction between the actual rootstock and scion. So we don't know exactly what happened there. We have to restructure our experiment to have higher survivorship and just be more careful with not letting them get infected. So unfortunately, no very conclusive results, but nonetheless, very interesting research. I really enjoyed it, yeah.
Thank you very much. This is very interesting. Do you think that the knocked out gene has anything to do with the high infection rates of the fungus?
Yeah, definitely. So if we're right in it being a knocked out gene, then its immune response will be basically turned off, and that could definitely explain why they all got infected and died. So we just have to be really careful with not letting them get infected, being more sterile, and also just increasing the scale of the experiment so that we would have more samples. Yeah.
Great. Thank you. That sounds like a good future plan.
Thank you.
Yeah. Thanks for the excellent work.